Hello students, I hope you all are well and in good health. Well, today in this video lecture we are going to study about the structure of bacteria. Okay, so in the previous lecture we have studied about the appendages, that is flagellum and the pili. In this lecture we are going to focus on structure. So first of all, let me draw a full diagram of bacteria, how it looks. Well, great. So what I'm drawing here is the cell membrane and mesosome and chromatophore. Well, let me first draw and then. So what I draw here, this is the plasma membrane or the cell membrane of the bacteria. Okay. Now this cell membrane has some infoldings, correct? Now see the first infolding that I made here, this one. This is mesosome. Now, in the previous lecture, we have seen that mesosome is analogous to mitochondria. Okay, so this is how mesosome look like. It is not necessary that it always has to be in this shape, but it is somewhat similar to it. Okay, great. Now, this is on an infolding of the cell membrane, right? You can see here it is just a infolding. After this infolding, the plasma membrane continues, and we find in some bacteria, not in all bacteria, okay, only in photosynthetic bacteria, that there is another infolding, and this infolding forms the chromatophore. This is analogous to chloroplast. We have again seen this all. And you can see that in the mesosome and the chromatophore, there is some folding like structure. It is highly folded structure. Great. Now, apart from this inner cell membrane, what else we find is inside this cell membrane, there is a fluid filled, and this fluid is called obviously cytoplasm or protoplast. Now, within this, we find there is DNA, correct? The genetic material. So here what I have drawn is the DNA, long double-stranded circular DNA, that is the chromosomal DNA of the bacteria. Apart from that, I am also drawing here is these pink colored circular structures. These are also DNA, but these are not the chromosomal DNA, but extra chromosomal DNA known as plasmids. Apart from that, here I am drawing these structures. These are ribosomes, and to be precise, these are 70S ribosomes. So there are ribosomes. Apart from ribosomes, again there are some granules. So the green ones are glycogen granules, and the other one are volutin granules. So apart from that, there are granules, and these volutin granules are storehouse of phosphate. So these are phosphate granules. Okay, so let me draw here, and now again I'm marking it. So green one is glycogen granules, and this is volutin granule. Now let me move it a bit. Well, so this volutin granule is metachromatic. What is the meaning of metachromatic? That it is multicolored. Okay, why we call it metachromatic? Because it can stain, it can bind to different dyes or different stains. As a result, the whole volutin granules can be colored to different dyes or to different colors, and these are used for staining of bacteria. Okay, what they are in function? In function, they are phosphate storage house. So these are the granules in which phosphate is stored, or these are granules of phosphate. Apart from this, see, I have already spoken of mesosomes, and you know it is analogous to mitochondria. Okay, these are plasmids. This is chromosomal DNA. Okay, and the chromosomal DNA is double-stranded and circular. Again, this is chromatophore and analogous to chloroplast. We have seen this. Now, these are the various organelles which are found in a prokaryotic cell, or in a bacteria, or I should say bacterium. Bacterium is singular, bacteria is plural. Okay, so never write bacterias. Bacteria is already plural. Bacterium is singular. So as I said, this entire place is filled with a fluid that is called cytoplasm, a protoplast. So I'm just filling it. So what you see here is once we fill it, okay, can you see that the cytoplasm is of different density here and there, correct? Can you see that around this chromosomal DNA, the cytoplasm or the fluid is much more viscous. It is jelly-like, very viscous, dense, right? So this area which is composed of this dense fluid-like material in which the chromosomal DNA is present, this whole area is called nucleoid. Okay, so this whole thing is called nucleoid. Fine. Can you see here now? So this thing is nucleoid and remaining is the cytoplasm. And now can you see that it looks like something, some organelle, it looks like a nucleus, but it is not nucleus. It is not distinct because it does not have a boundary. It does not have nuclear envelope. It does not have nuclear membrane. Okay. Let me tell you that outer and inner nuclear membrane both together called nuclear envelope. Great. So this is nucleoid. I think now you are able to appreciate it, the difference between nucleoid and true nucleus. So this is nucleoid which has this chromosomal DNA. So this is how the inside a bacteria looks like. Now apart from this, outside the cell membrane, what we find is a layer of cell wall, and this cell wall is made up of peptidoglycan. Now the space between this cell membrane and cell wall is called periplasmic space, correct? We have seen it. So we have seen that there is a space between cell membrane and cell wall that is called periplasmic space. Well, now after this what we find is another layer which I have drawn here in blue. This is called glycocalyx. Remember, this is glycocalyx. So glycocalyx is the outermost layer. Inside the glycolysis we find cell wall, inner to cell wall, cell membrane. So these are three coverings in a bacterium. Great. Apart from this, if you see from outside, you find that a bacterium may have flagella or flagellum, which I have drawn here, okay, as well as it can have the other appendage as pili as well. Can you see? So this is the complete structure of a bacteria. What we have seen, that the outermost layer is glycocalyx, then is cell wall, and then is cell membrane. Now cell membrane may show infolding to form mesosome as well as the chromatophore. Inside the cell membrane we find the cytoplasm or protoplast. In the protoplast we find in the center there is nucleoid, which is dense fluid-like structure containing the chromosomal DNA, and this chromosomal DNA is double-stranded and circular. Apart from this, there are some extracellular DNA, extrachromosomal DNA called plasmids. Okay, and in the cytoplasm we find some granules like glycogen granules, volutin granules, and 70S ribosomes. Great. So this is how structure of bacteria appears, or this is how bacteria looks like. Now let us see one by one each structure. Now let's start with flagella. Now flagella we have already seen in the previous lecture in detail, right? So I hope you do not have any doubt about flagella. So flagella we have seen. We also have seen or studied pili. So flagella and pili I'm putting tick to. The third layer from outer side is glycocalyx. So let's see what glycocalyx is. Now, it is the outermost slime layer. What is its function? It protects the bacteria from WBCs. WBCs, that is white blood cells. So this glycocalyx is the outermost protective layer of the bacteria, which mainly protects it from the WBC. That is, it helps in evading the host immune system. Now understand, what is this immune system? देखिए, हम लोगों में किसी भी पैथोजन से लड़ने के लिए एक सिस्टम होता है, एक पूरा का पूरा organized system है, that is called immune system, and this immune system is composed of the WBCs. Okay, white blood cells जो हैं आपके mainly neutrophils, acidophils, या फिर आपके lymphocytes, okay, macrophages. So ये सारे जो different natural killer cells, ये सारे different type of WBCs हैं जो कि एक साथ मिल करके आपकी बॉडी के डिफेंस सिस्टम की तरह काम करते हैं, जिसको हम इम्यून सिस्टम कहते हैं. ठीक है. Now, this immune system is responsible for fighting against any kind of foreign particle that comes inside your body or any kind of pathogen that comes inside your body and tries to harm your body. Okay, so this is the work of immune system. The property is called immunity. Okay, so the immune system provides you immunity against the pathogens. How? They target these bacteria or any pathogen. How they target? They target by the help of the presence of molecules, chemical molecules on their cell wall. So this immune system know how to recognize the bacteria. Okay. Now what happens if above this cell wall there is a layer of glycocalyx? It will not let the molecules of cell wall to be exposed to your immune system or WBCs, and thus it protects it from your WBCs, right? So that the bacteria will have an advantage, correct? So that it can invade your system. It can take hold of your system. It can make you sick, make you diseased. But आप diseased हो रहे हो, bacteria को तो फायदा हो रहा है ना, right? He is winning the battle, and glycocalyx is helping them. Let's see. So what it is doing, it is increasing the pathogenicity of the bacteria. Can you understand? If there is two bacteria, same bacteria, hypothetical situation, okay, two same kind of bacteria, one with glycocalyx, another without glycocalyx. So obviously one without glycocalyx will be effectively targeted by your WBC and will be thrown away. So it will not be that pathogenic, correct? Why I'm saying not that pathogenic? Because in some case it may become pathogenic when your WBCs are not working that well, or if the bacteria is in very large quantity. In either case, even without glycocalyx, they can make you fall sick, or they can defeat your immune system, right? One or the other, same thing. In case the bacterium has glycocalyx layer, what will happen? This glycocalyx will not allow this WBC to interact with this bacteria to identify this bacteria, and thus these bacteria will be having better chances to infect you, to make you fall sick. That is, their ability or their pathogenicity is higher. So what glycocalyx is doing, it is increasing the pathogenicity. याद रखिएगा इस चीज को. Now, the glycocalyx is of two types. Okay, one is called slime layer, the other one is called capsule. Now, this is on the basis of the structure of glycocalyx. Okay, so structure and the composition of glycocalyx. The glycocalyx is divided into two types, that is slime layer or capsule. Now, what is slime layer? Again, slime, चिपचिपा जैसा चीज, and layer, a thin layer. Correct? So glycocalyx, a type of glycocalyx which is slime layer. In case of slime layer, it is loose thin layer of polysaccharide only. याद रखिएगा, only. So if as a glycocalyx there is a loose thin layer of polysaccharide only, we call such glycocalyx slime layer. Okay. If the other case, if it is thick layer of polysaccharide and polypeptide, then we call it capsule. समझ में आ रहा? So अगर thick layer है और thick layer होगा तो दो चीजों का बना है, पॉलीसैकेराइड एज वेल एज पॉलीपेप्टाइड. पॉलीपेप्टाइड मतलब प्रोटीन्स. ओके, तो थिक लेयर को हम क्या बोलते हैं? कैप्सूल. ये प्रोटीन और पॉलीसैकेराइड यानी कि कार्बोहाइड्रेट दोनों का बना है. जबकि जो स्लाइम लेयर है वो मेनली किसका बना है? सिर्फ पॉलीसैकेराइड का. और दिस इज थिन एंड लूज. सो स्लाइम लेयर एंड कैप्सूल, याद रखिएगा इस चीज को. सो ग्लाइकोकैलिक्स एक और काम करता है कि ग्लाइकोकैलिक्स हेल्प्स इन कॉलोनी फॉर्मेशन. क्यों? जो ग्लाइकोकैलिक्स है वो सेल टू सेल एडिशन में हेल्प करता है. एंड जब बैक्टीरिया ग्रुप में होंगे या एक जगह पे बहुत सारे बैक्टीरिया होंगे तो ये जो ग्लाइकोकैलिक्स है इनसे ये अटैच होकर के दे स्टार्ट टू फॉर्म अ कॉलोनी. ग्रेट. सो दिस इज ग्लाइकोकैलिक्स. नाउ लेट्स मूव टू द नेक्स्ट कंपोनेंट, दैट इज सेल वॉल. ओके, याद कीजिए ग्लाइकोकैलिक्स के अंदर क्या था? सेल वॉल. सो वी आर मूविंग टू सेल वॉल. नाउ व्हाट इज सेल वॉल? इट इज आउटर प्रोटेक्टिव कवरिंग. समझ में आ रहा है? It is outer protective covering of bacteria. Now, in case of bacteria, the cell wall is composed of peptidoglycan. याद रखिएगा, basic question है. ठीक है? Peptidoglycan. Now, again, what is peptidoglycan? पहले भी हमने बात किया है, आज फिर बात कर रहा हूं. देखिए, दो वर्ड से ये बना हुआ है, पेप्टिडो एंड ग्लाइकन. पेप्टिडो का मतलब हो गया प्रोटीन और ग्लाइकन का मतलब हो गया कार्बोहाइड्रेट. तो पेप्टिडोग्लाइकन जो है एक हाइब्रिड मॉलिक्यूल है. मतलब एक ऐसा मॉलिक्यूल है जिसमें कि आधा पार्ट प्रोटीन है और आधा पार्ट कार्बोहाइड्रेट है. नो, आई एम सेइंग आधा, इट इज नॉट एग्जैक्टली हाफ हाफ. व्हाट आई एम ट्राइंग टू से, एक ऐसा मॉलिक्यूल है जिसमें कुछ पार्ट प्रोटीन है और कुछ पार्ट कार्बोहाइड्रेट है. सो सच अ मॉलिक्यूल इज कॉल्ड पेप्टिडोग्लाइकन. ठीक है? और बैक्टीरिया का या किसी भी प्रोकैरियोटिक ऑर्गेनिज्म का जो सेल वॉल होता है, इट इज कंपोज्ड ऑफ पेप्टिडोग्लाइकन. रिमेंबर ना? सो ये किस तरह का मॉलिक्यूल हो गया? इट इज अ कॉन्जुगेट मॉलिक्यूल. करेक्ट? वेरी वेल. नाउ द बैक्टीरिया on the basis of cell wall can be divided into gram positive and gram negative. Now, in case of gram positive, right, let us see what we will do is we will do a comparative study. ठीक है? So gram positive के केस में क्या होगा? It has thick single layered cell wall which is composed of only peptidoglycan layer. ठीक है? याद रखिएगा. The bacteria or bacterium having a thick single layered cell wall composed of only peptidoglycan is called or is gram positive. Okay? Now, this peptidoglycan layer is made up of obviously peptidoglycan, and chiefly this peptidoglycan is murine. Okay, so the name of this peptidoglycan molecule is murine. याद रखिएगा इसको. Now, murine in case of gram positive bacteria is around 70 to 80% of the cell wall, right? So 70 to 80% of the cell wall is composed of murine, a type of peptidoglycan in case of gram-positive bacteria. The remaining amount is mostly lipid. So lipid is low in quantity. Correct? Secondly, what we see in case of gram-positive, the thickness, right? We say it is thick. How much? It is around 20 to 80 nanometers thick. So it is quite thick. Just to give a reference, the plasma membrane, the thickness of plasma membrane is only 7.5 nanometer. Okay, so plasma membrane thickness is 7.5 nanometer, but the thickness of gram-positive bacteria cell wall is 20 to 80 nanometer. Okay, great. Now let's see something about gram-negative. Now, the gram-negative bacteria has thin cell wall. Okay, the thickness is low. It is thin, less thicker, thin cell wall, but it is double layered. Can you see the irony here? The thicker cell wall is single layered, but the thinner cell wall is double layered. In case of gram-negative bacteria, the cell wall is thin but double layered, and these two double layers, the inner layer is made up of peptidoglycan, okay, that is mainly murine, and since it is very thin, the overall composition of murine in this type of cell wall is only 10 to 20%. Okay, great, only 10 to 20% in whole of the cell wall. Now, if you compare the outer layer is made up of lipopolysaccharide. Now, what is lipopolysaccharide? First of all, again, it is of two words. It is made up of two words, lipo and polysaccharide. Polysaccharide is carbohydrate. Lipo is for lipid. So again, it is a conjugate molecule which is made by the conjugation of lipid molecule with the carbohydrate. So the outer layer is made up of lipopolysaccharide. So in a gram-negative bacteria, the cell wall is thin, but it is split into two layers. Inner layer, peptidoglycan, outer layer, lipopolysaccharide. Okay. Therefore, the lipid content is comparatively higher. Now, how thick this membrane, this cell wall is? Only about 8 to 12 nanometers. Can you see? It is significantly thinner, 8 to 12 nanometer thick. Apart from this, see what are other differences? The basal body of the flagellum in case of gram-positive bacteria is composed of only two rings. याद कीजिए जब हम bacterial flagella पिछले क्लास में देख रहे थे, we have seen that the basal body has four rings, right? L, P, S and M. जबकि अगर ये gram positive bacteria है, so its basal body will contain only two rings and those are S and M. So S and M rings are found in, can you answer? Yes, in the cell membrane, right? L and P were found in the cell wall. Great. Now, if we are talking about gram-negative, so in case of gram-negative bacteria, the basal body of the flagellum contains all the four rings. So the structure we studied of bacterial flagella was actually of gram-negative bacteria, right? It has all the four rings, that is L, P, S and M rings. Good. Now, apart from that, what we see in case of gram-positive bacteria, only a few pathogenic bacteria are gram-positive. Okay, only a few pathogenic strains are gram-positive, whereas most of the pathogenic bacteria are gram-negative. Okay, they are gram-negative. Secondly, last but not the least, the most important one rather, on what basis we call gram-positive or negative is not the thickness of the cell wall. Rather, we call a bacteria gram-positive or gram-negative on the basis of gram staining. Okay, now what is this gram staining? We will see later in this chapter. Now, It is a method of coloring the bacteria which was given by a scientist called Hans Christian Gram. Okay, Hans Christian Gram, HC Gram, and hence in his honor we call it Gram staining. The bacteria which retains the gram stain are called gram positive. Those who do not are called gram negative. Okay, so all the bacteria which retains the gram stain and appear purple or violet upon gram staining are called gram positive, while the bacteria which do not retain the gram stain upon gram staining are called gram negative. So what they appear? They will appear pink or red. And why it is so? It is so because of the color of the counter stain. Okay, तो जो कलर नहीं लेते हैं, ग्राम स्टेन को नहीं पकड़ते हैं, उनको बाद में हमें काउंटर स्टेन होता है रेड कलर का या ऑरेंज कलर का, उससे हम करते हैं. सो उससे कलर करने के बाद ये पिंकिश रेड या रेड जैसा दिखता है. तो ग्राम नेगेटिव बैक्टीरिया अपीयर्स पिंक और रेड इन ग्राम स्टेनिंग. Okay, great. So this is a broad difference between gram positive and gram negative bacteria. Great. Now one more thing I would like to add here is the gram negative bacteria are also called L form or Lister form. Okay, why it is so? Because this L represents the lysozyme. Okay, all these gram negative bacteria, they are sensitive to lysozyme. Lysozyme is a kind of enzyme. This enzyme targets the bacterial cell wall. Okay, and which kind of cell wall? The gram negative cell wall, and it dissolves the gram negative cell wall. So if a gram negative bacteria has been treated with lysozyme, its cell wall will be dissolved by this enzyme, and a bacteria without cell wall will be left. Correct? Now this bacteria without cell wall is called Lister form or L form. Okay. Now this bacteria without cell wall is very sensitive and it dies. That's why lysozyme is used as an antibacterial agent. Okay. Now you will be surprised to know that lysozyme is found in our saliva, is present in our tears, and that's why a bacteria when goes into our eyes or in our mouth, in the presence of lysozyme, they are killed or they are killed by the lysozyme. Okay, so this was glycocalyx and cell wall. Very well. So this is all for today. In next video lecture, we will study about the gram staining and further or the remaining structures of bacteria. Till then, stay tuned and stay safe. Thank you.